we'll just go ahead and get to going. Last week, it was Wednesday when we last lecture and first talked about this. We learned that there were three different processes involved with the formation of urine. Okay. The first of which is called glomerular filtration. It involves that glomerulus, the massive capillaries that's found inside of our renal corpuscle, or as part of our renal corpuscle, I should say, found inside of the capsule, where we filter out a lot of the stuff in our blood, basically the water and all the small stuff. Okay. From there, what we call filtrate will funnel through our renal tubule. We'll then either reabsorb it back into the blood, or we'll secrete stuff into the tubule from the blood. Okay. And ultimately what's formed is urine, which will then get carried to our bladder and then carried out of the body. We'll talk just briefly about that because those processes are pretty simple. So we'll spend the majority, like I said the other day, talking about the kidneys. So let's talk more about glomerular filtration. We've actually talked about filtration before. Okay, this was something that was introduced to you back when we talked about blood vessels. Okay. But the principle of filtration being the use of pressure to force fluids and solutes through a membrane, is the same in our glomerular capillaries as it is in capillaries elsewhere in the body. However, the volume of fluid by the renal corpuscle is much larger than in the other capillaries because of kind of three things. And first thing, glomerular capillaries have a very large surface area available for filtration because they're very long and extensive. The filtration membrane, which is what we talked about on this slide, is very, very leaky. We'll just call it that. Okay. First of all, the capillaries are fenestrated, so they have those holes, making them like 50 times leakier than other capillaries. And then the two membranes outside of it, and then the cells, allow for easy passage of certain materials. Okay. So filtration membrane, very thin and porous. The capillaries, like I said, are about 50 times leakier than most capillaries everywhere else because of those large fenestrations. And then the blood pressure in those capillaries is very high because of the differences in size with the afferent and the efferent arterioles. And that's what we can show here on the next slide. Okay. Go ahead and write down what you need to write down on this page, and I'm going to explain what I mean, and then I'll talk about the other stuff that's on this page. Okay. So here's basically the same figure that we saw the other day of our glomerular capsule, the glomerulus, the afferent arteriole, and the efferent arteriole. Okay. Reading through, glomerular capillary blood pressure is high due to the efferent arteriole being smaller in diameter than the afferent arteriole, which causes resistance to the outflow of blood from the glomerulus to be very high. It's not very drastic, but the width of this afferent arteriole, which is the arteriole that brings blood into the corpuscle, is actually wider than this efferent arteriole, which is what brings blood out of the corpuscle. Okay? So basically, more can come in than what can leave, which means that the blood in here is under a lot of pressure. Speaking of that pressure, what I'm describing is actually the first of the three pressures that affects glomerular filtration. Okay. That first pressure is called GBHP, or glomerular blood hydrostatic pressure, and it's the only one that promotes filtration. Okay. This is the pressure in the glomerular capillaries, and it's going to promote filtration by trying to force water and solutes through the filtration membrane out into the space. The other two pressures are represented with the purple arrows and the blue arrows, as you can see point inwards, oppose filtration. We've got capsular hydrostatic pressure, which is the pressure inside the capsule due to a fluid that's already there. And then blood colloid osmotic pressure is pressure within the capillaries that avoids or opposes filtration due to the presence of proteins. So it tries to keep stuff in the blood. Okay. Now, you can take those three pressures and calculate whether or not filtration is going to occur. And the calculation is NFP, short for net filtration pressure, equal to GBHP minus CHP minus BCOP. Are you going to need to do any calculations on the test? No. But as you can see, when we actually apply some numbers to it, 55 minus 15 minus 30 ends up as a positive number. I mentioned this back when we talked about blood vessels, but a positive NFP promotes filtration. So as long as that number ends up positive, we're going to form filtrate. If that number ends up negative, filtration will not occur. Now, the importance of NFP is demonstrated on the next slide. When we're talking about the rate at which we form filtrate. A concept called glomerular filtration rate, or GFR. GFR, as I said, refers to the amount of filtrate formed, not just one corpuscle, but all the corpuscles of our nephrons in both of our kidneys each minute. So it's the amount of filtrate we're making total between the two kidneys. Now, listen to these numbers. Average is 105 mils per minute in women, 125 mils per minute in men, which adds up to 180 liters, or about 45 gallons of filtrate formed per day. That's how much stuff we pull out of the blood and begin the formation of urine. Now, do we pee 180 liters of urine a day? If you say yes, you need to go back and figure out what a liter is. Okay, or you need to go to the doctor. Okay. Obviously, you can't. That's impossible. So what that tells us is, and that's stated right here, most of the glomerular filtrate must be returned to our blood and recycled. And we're going to talk about that today. Homeostasis of body fluids requires that our kidneys maintain a relatively constant GFR. So we need to constantly be making filtrate. Okay? If it's too high, needed substances may pass so quickly that renal tubules, through the renal tubules so that they might be lost in our urine. If it's too low, we'll not lose enough stuff. And then certain waste products might not be lost through our urine, and then things can start to go wrong because wastes should not be building up in our blood. Okay. As I said on the previous page, NFP affects GFR. Okay. They're directly related. Any change in NFP will affect GFR. Okay. So we can adjust NFP, which therefore affects or adjusts GFR. And there are three mechanisms that will control GFR by either adjusting flow into and out of the glomerulus or by altering the capillary surface area available for filtration within the glomerulus. Okay. The first thing we're going to talk about is autoregulation. You guys remember what autoregulation is? We've talked about that word before. Self-regulate. Okay, it's where our kidneys can self-regulate. Okay, neural, and it's going to involve that sympathetic nervous system that we can't avoid. We've talked about for so many stinking lectures now. Okay, that fight or fight mechanism. And then hormones and how they regulate. And some of these hormones, actually all of these hormones we've talked about before, so a lot of this should not be surprising brand new information. Well, let's first talk about autoregulation. Renal autoregulation is the ability of our nephrons to adjust their own blood flow and GFR without external control. So without nervous or hormonal control. Okay. This allows them to maintain a stable GFR in spite, and I should say, of changes in arterial blood pressure. So even if we have great changes in blood pressure throughout the rest of the body, our GFR is still able to be maintained. Okay. The first of the two mechanisms is called the myogenic mechanism. Okay. This involves smooth muscle. Myo, remember, means muscle. Okay. So this occurs when smooth muscle that surrounds the afferent arteriole contracts because it was stretched when blood pressure was increased. This contraction of the afferent arteriole will cause vasoconstriction, which means less blood comes into the corpuscle. If there's less blood coming in, what's going to happen to filtrate? Is it going to go up or down? There's less blood to be filtered, so there's less filtrate going to be formed. Okay? So, this reduces GFR to its previous level. And that's what we're showing off with this figure here on the next page. Okay. We have normal GFR, but some disturbance causes blood pressure to go up, which causes GFR to go up. Okay? But we don't want that to occur. So we want GFR to remain normal, right? We just said that. Okay? So, we detect that increased arterial pressure because there are stretch-sensitive receptors in those afferent arterioles, okay, resulting in calcium inflow into smooth muscle fibers. We know how important calcium is for muscle contraction. More calcium means a stronger contraction. Stronger contraction causes vasoconstriction. Less blood flow in, less GFR can be formed. Therefore, GFR drops, returns us to normal. So what type of feedback are we demonstrating here? Negative. Good. Okay. Now, when blood pressure drops, the opposite of this is all going to occur. Okay? Smooth muscle cells are stretched less, allows them to relax, afferent arterioles dilate, therefore GFR goes up. Okay? This myogenic mechanism normalizes renal blood flow and GFR within seconds after a change in arterial blood pressure or systemic blood pressure has occurred. Now, tubuloglomerular feedback is going to describe that conveyor belt analogy that I was talking about the other day when making Barbie dolls. Okay, remember how I talked about if the conveyor belt's going way too fast, there's going to be eventually a point where somebody's not going to be able to keep up, like putting the Barbie doll heads on top of the Barbie doll. Okay. Well, if our conveyor belt, in this case, the speed in which GFR is being formed is too high, we're not going to be able to reabsorb enough solutes, enough water in our proximal convoluted tubule and our loop of Henle. Okay. We have cells of the macula densa that detect those chemicals, and in response will secrete chemical messengers that do two things. They're going to vasoconstrict the afferent arteriole, and they're going to send chemicals that inhibit any vasodilation of that afferent arteriole. Okay, so that causes GFR in the same nephron to decrease. Okay, when blood pressure falls, causing GFR to be lower than normal, opposite sequence of events occurs, although to a lesser degree. Okay. Tubuloglomerular feedback does operate more slowly than the myogenic mechanism. So let me go back to our figure here of our nephron. Okay, remember I had you guys kind of identify this region right here of the distal convoluted tubule. Okay, I described that as the juxtaglomerular apparatus, which is where the tubule passes right by that afferent arteriole. Okay. Let's say that our conveyor belt's moving way too fast. Everything's passing way too fast, and we're not able to pull back in enough stuff, or the stuff that we want. There are cells here that can say, hey, we need to slow things down, so they hit a switch. The switch slows the amount of blood in by causing vasoconstriction here, which means less blood flow in, things can slow down. Does that make sense? Okay, and that's how it works. Here's the feedback loop to describe this. Okay. So stimulus causes a disruption in GFR. We've increased it. Okay, so our conveyor belt's going way too fast. The cells of the macula densa detect changes. This time we're looking at chloride, sodium, and water, so salt and water balance. We're not pulling enough salt and water back into the blood, so they're going to send signals over here to the blood vessel. They cause them to constrict, less blood in, decreases GFR, so we are peeing out too much salt and too much water, and we can help retain it. Now, what type of feedback is this demonstrating? This also demonstrates negative feedback, and you can see that right there. Okay, so that's autoregulation. Let's look at neural. Neuroregulation is by way of the sympathetic nervous system fibers secreting norepinephrine. We've learned previously that when norepinephrine is released, it's going to cause vasoconstriction of arterioles in our kidneys, especially those afferent arterioles. Well, what response does the sympathetic nervous system do or produce? What's the name of that response? What's the fight or flight? Okay. And that's what we do like in a life or death situation or like when we're exercising, right? Think of it as simple as this. When we're running away from a bear, do you think we give two F's about making urine? No. What do we need to do? Well, yes. And that means we need to get blood flow to the working muscle, right? Okay. We also need to have as much blood as possible, okay, because we're going to start sweating, and that will reduce our blood volume. Okay. So we don't want to compound that problem by peeing out what water we do have, because we need it to make sweat. And we also want as much blood as possible, because more blood means more oxygen delivery to our muscle. Okay. So at rest, like you guys are all sitting right now, I would assume your sympathetic activity is very low. Okay. So therefore, your afferent and efferent arterioles should be pretty well dilated, and therefore GFR is going to be regulated by that autoregulation. Okay, so there's really no influence at rest. Okay. With moderate sympathetic stimulation, both the afferent and efferent arterioles constrict to the same degree, causing a slight decrease in GFR. And then great or high levels of sympathetic stimulation, like during a hemorrhage or during exercise, will cause great constrictions of the afferent and efferent arterioles. Actually, a little bit more of one, but not really too important. Therefore, blood flows less into the nephrons. Therefore, GFR drops significantly. Because, like I said, we don't care about making urine if we're being chased by a bear, or if we're running a 1,600-meter race or something like that. All right, let's now talk about hormones and how they regulate GFR. Two hormones help to regulate GFR. Let's first talk about atrial natriuretic peptide. We first introduced this back when we talked about blood vessels. We didn't actually talk about this hormone at all back when we talked about the endocrine system. But we learned that this hormone was produced by our heart, specifically the atria. That's why it's named atrial natriuretic peptide. And we secrete it when our blood volume is elevated. So a great way to reduce blood volume is by peeing out water. So this hormone is going to increase GFR. It does so by increasing the capillary surface available for filtration by relaxing what we call the glomerular mesenchymal cells. Let me go back to show you those cells again. Here's our picture of the renal corpuscle. Okay, here's our glomerular capillaries. In between those capillaries, you can't really see them very well, but there are these mesenchymal cells. These guys can control how much surface area is available for filtration. Okay, they relax, and that basically means there's more ability to form filtrate. And that means filtration is going to go up. Okay. Now, the other hormone is actually a series of hormones and enzymes, chemicals, part of a mechanism or system that we've mentioned a little bit before, the renin-angiotensin-aldosterone mechanism. The original stimulus for this whole process is a drop in blood pressure. Okay. And so the overall goal, since this is going to be related to or regulated by negative feedback, is to increase blood pressure. And everything that occurs here is going to increase blood pressure. either directly or indirectly. Okay. So when systemic blood pressure drops, our sympathetic nervous system will send signals in cells of the juxtaglomeralar glomerular apparatus to secrete the enzyme renin. Renin is the enzyme that interacts with the plasma protein angiotensinogen, causing releasing a chain of amino acids called angiotensin 1, which is then converted to angiotensin 2. Angiotensin 2 is the hormone that actually does these various actions. First of all, it causes vasoconstriction. So that right away directly causes an increase in blood pressure. Okay. It constricts both the afferent and efferent arterioles, reducing GFR and therefore water loss. So if water volume goes up, what happens to blood pressure? It goes up, right? Because there's more water in the plasma, therefore blood volume goes up, which causes blood pressure to go up. Okay. We stimulate sodium chloride and water reabsorption. What's sodium chloride best known as? Salt. So we retain salt and we retain water. Blood volume goes up. What's that going to do to blood pressure? Goes up. Okay, seeing a trend here, right? Okay. It also stimulates the release of aldosterone from the adrenal cortex. What's the nickname for aldosterone? Salt retaining hormone, just like here. It's going to promote reabsorption of salt, but also water, increasing blood volume, therefore increasing blood pressure. Stimulates the secretion of antidiuretic hormone, ADH, released by the posterior pituitary, helps us retain water, increases blood volume, therefore increases blood pressure. And then the final thing is it stimulates the sense of thirst. It encourages water intake, which increases blood volume, which therefore increases blood pressure. Okay. So we do a lot of things that cause increases in blood pressure. Now, this is great because there's a drug out there, or a series of drugs that can be taken by people with hypertension or high blood pressure. Anybody ever heard of ACE inhibitors before? ACE inhibitors, the ACE is short for angiotensin converting enzyme. Prevents this from being converted to this, which prevents all this, which do a whole lot of increasing in blood pressure. So if you ever heard of that drug, now you know what it does. Okay. Table 26.2 is just the same, basically, series of things that we just talked about, so you can use it to study or not, whichever. All right, so that's the first step. The second two steps are, once again, reabsorption and secretion. Well, we've already defined both of those, but remember, tubular reabsorption is the process of reclaiming water and solutes from our tubule fluid and then returning them to the blood. It occurs in all three segments, and even the collecting ducts. Most of the reabsorption, especially of the water, takes place in our proximal convoluted tubule. Okay. And they are going to be returned to our blood by two different routes, a transcellular route and then a paracellular route. Okay. Do you guys remember what the prefix trans means? Yeah, exactly. And that's why the word through is bolded here. Okay. Across or through is what trans means. This is where a substance passes directly through the cell from one side and then out the other side. Whereas paracellular is where they pass between cells. And that's, I drew it over there on the board. Put the figure here on the next page. I kind of forgot that that figure was on here. It's probably a little bit easier to see. Okay, so paracellular is where substances pass kind of between two cells. Okay, here's our tubule lumen, so this is inside of the tubule. Here are the cells that line the tubule. You can see they have microvilli. And then here's the interstitial space, and then here's our peritubular capillary, the blood vessels for which stuff gets brought back into the blood. We can pass between cells, or we can just go right through cells, through the apical membrane and then out through the basolateral membrane. Okay. Make sense? So let's talk now about how sodium is a very important chemical for reabsorption. Not just for sodium, but for other things. Okay, so reabsorption is primarily coupled to movements of sodium Na+, which is actively transported out of the tubular cell and into the interstitial fluid and blood plasma by a sodium-potassium pump located in the basolateral membrane of the tube cell. That's what we show right here. Okay. So we know active transport requires ATP, and when we pump something, we go which direction? Down or against a gradient? Not down, but against. Okay. So instead of going from high to low, we go from low to high. So what little sodium is in here gets pumped out here, which means that this cell or these cells are going to be very, very low in sodium at all times. Which means, and follow me here, if we're low here and we're high here, what are we going to try to do? Bring sodium in. Down its gradient. Okay. So we're always moving sodium here, but to keep this cell layer or this cell low in sodium, we are pumping the sodium out. Now, sodium always is going to want to come in for that reason. And so things like glucose can take advantage of that. Glucose is going to get co-transported with sodium into the cell through a sodium-glucose symporter. So glucose is going to take a piggyback ride into the cell, and then from there it can get pulled back into the blood. Here's facilitated diffusion, which is not that important, but that's what happens before it then diffuses into the blood. Does that make sense? Hydrogen ion also utilizes sodium, but in a different way. And that's what we can see here on this page. We counter-transport sodium, or hydrogen ion, out of the cell and into the lumen in exchange for sodium via an antiporter. Okay, symporters pull two things in the same direction. Antiporters pull two things in opposite direction. Here's hydrogen ion leaving the cell and entering the tubule lumen. Sodium is coming in. So hydrogen ion in this case is being secreted, not reabsorbed. Okay, because that's what secretion is, leaving and entering into the lumen of the tubule. Okay. Now, where does that hydrogen ion come from? Coming from the same reaction that we've seen for the last several weeks. When CO2 accumulates in the tubule cell, it's going to combine with water. Remember, Ca is short for carbonic anhydrase, the enzyme that joins those two things together, forming carbonic acid, which then dissociates to bicarbonate and hydrogen ion. Boom, there's the hydrogen ion that I was just talking about. Okay. Most of the bicarbonate, HCO3, is reabsorbed by the proximal convoluted tubule by facilitated diffusion, which then helps supply the body with an important pH buffer. It's going from the tubule cell into the blood, can become a buffer, and we'll talk about that more at the end of the lecture. Okay. The hydrogen ion, after it's secreted out, okay, is going to basically do the exact opposite that it did inside the cell. With any bicarbonate that's in the tubule lumen, it will combine with it to form the acid, and then we're going the other way to form water and CO2. CO2 will get reabsorbed, water will stay out. Okay. So that's how the hydrogen ion actually leaves our body. It's actually combined together with oxygen to form water. Okay. But that helps demonstrate the fact that we can maintain pH balance or acid-base balance by pushing hydrogen ions into our urine. And that's why if you take a sample of blood and you put a pH strip on it or drop a urine on the pH strip, it should read acidic. Now, down at the bottom is where we can show solute and water reabsorption. Solutes like chloride, calcium, magnesium, and urea move, and they're going to pull water with it. Remember at the beginning of the semester how we talked about if you drink salt water, you're going to become dehydrated because it's going to suck all the water out of your cells to go where the salt is. Okay? So when chloride and these other ions move into the cells, water is going to chase it. Okay? Water moves by way of osmosis. Okay? And then if they move from the cell into the interstitial space, water is going to chase it. And then if they move from the interstitial space into the blood, guess who's going to chase it? Water. So water is always kind of like two or a step behind, but it's always chasing it. And so that's how we reabsorb water. And like I said, in that proximal convoluted tubule, we reabsorb like 99% of that water that we just filtered out. So that's the first segment of our tubule. Let's now talk about the loop of Henle, the second segment. Reading through, because the proximal convoluted tubule reabsorbs about 65% of the filtered water, fluid that enters the loop of Henle will have, I should not say will have a very different chemical composition than it was entering the proximal convoluted tubule. Loop of Henle continues the reabsorption of water, about 15%, and those same solutes, but for the first time, water is not coupled to the movement of those solutes because part of the loop is relatively impermeable to water. Remember, impermeable means water can't pass through. Okay? That's because the thick segment doesn't like water passing through it. Okay? And remember, there are thick and thin segments of the loop of Henle. We can see those here on this page. You can see the thick segment here in the ascending limb, more thinner parts of the descending, and then the part of the ascending limb. Okay? Look what we're doing to water in the descending limb. Look what we're doing to sodium chloride in the ascending limb. Those two limbs affect one another. Specifically, this limb, what we're doing here affects this limb. For that reason, we call this a countercurrent multiplier mechanism. Okay. And this helps to increase the concentration of the renal medulla. Renal medulla, remember, is the middle part of a kidney, which is where the loop of Henle is predominantly found. Okay. With water, what we're doing is reabsorbing it in this descending limb. Why? Because the fluid in the loop of Henle is like the ocean water. It's really salty because of what we're doing in this ascending limb. We're pumping out sodium chloride, aka salt. Okay, so this is just like salt water. They say if you taste the middle part of the kidney, it's going to taste really salty. Okay. Now, any of that water that gets pulled out in this descending limb is going to get pulled right into the bloodstream by way of what's called the vasa recta. The vasa recta is the blood vessels that surrounds the loop of Henle. You can't really see it. But if I went back to the picture of the nephron, you'd be able to see it. Okay. So we can describe the renal medulla as very hypertonic. Hypertonic was like salt water, really, really loaded with solutes. Okay. Presence of blood vessels or vasa recta surrounding the loop of Henle allows water to be carried away from the loop, which further contributes to the hypertonic environment. Urea is also picked up from the interstitial fluid in the descending limb. So we're performing secretion of urea, and we can see that right here. Now, what happens to urea is called recycling. Okay, we pick it up here in the lumen of the tubule. It goes through and then back down the collecting duct, which happens to pass right by that loop of Henle, and look what we do with it here. We're reabsorbing it. So we secrete it, and then we end up reabsorbing it, and then it secretes again. So that's why we kind of call it a recycling. Now, is urea going to get constantly stuck in that recycling? No, eventually some of it will get lost because we're not going to reabsorb all of it. So a lot of it actually will end up being pulled out in our urine. So eventually that's what's going to happen. So that's why we call that urea recycling. Okay. I already talked about this, but sodium and chloride are reabsorbed from the filtrate in the ascending limb, where the reabsorption of salt makes the tubular fluid very diluted or hypotonic. Okay. This creates and maintains a high osmotic pressure in the medulla's interstitial fluid to promote osmosis of water, what we were just talking about. Okay. Sodium chloride is moved into the surrounding interstitial fluid of the renal medulla due to active pumping of the thick sediment. And that's what we're showing right here. Okay. Sodium chloride coming into the cell, and then active transport tells you that it requires ATP to pump it out into that interstitial space. Does that make sense? Pretty complicated what's happening, but it's pretty cool what's happening. All right, our last segments of the nephron are the distal convoluted tubule and the collecting ducts. That's what DCT and CD stand for. If you can't remember that, might not be a bad idea to write that somewhere near this slide. DCT is short for distal convoluted tubule. CD is short for collecting duct. Some of those same hormones we talked about just a little bit ago with regards to GFR also affect reabsorption and secretion in these last parts of our nephron. You guys finish writing, and we'll talk about it. All right, so we just talked about this, but since salt is pumped out of the filtrate in the ascending loop, the fluid in the distal convoluted tubule becomes very hypotonic. It's really diluted at this point. It's really watery, not a lot of solutes in it. Okay? Whereas the interstitial space, or the interstitial fluid in that interstitial space of the renal medulla is going to be very hypertonic. It's really salty because of that pumping aspect that we just talked about. Okay? This helps form the osmotic drive for the reabsorption of water across the walls of the collecting ducts, allowing for some water conservation. Okay. So this does actually occur as well in the DCT, but a lot of this happens in the collecting duct. Okay. And we can see that here in our figure on that previous page. Here's our DCT. You can see water is being reabsorbed. Okay. In the collecting duct, we also see water being reabsorbed. This time it's due to hormones, okay? Or some of it's due to hormones. Okay? Now, these numbers I didn't really talk about, but they are indicative of the osmotic drive, or the pressures pushing osmosis to occur. And I don't really want to explain it, it's kind of complicated. So let's talk about the hormones that are going to stimulate principal cells. Okay. Of the first 4 hormones, only one of them is going to promote water secretion. The others all promote water and possibly salt reabsorption. The only one that promotes water secretion is ANP, atrial natriuretic peptide. That's the one that also increased GFR, because that's trying to reduce blood volume. It's wanting us to get rid of more water from our blood. ANP excretes more salt and more water in our urine, causing reduction in blood volume and blood pressure. The others are all geared towards reabsorption of water or salt, which then affects water. Because like I said, if you retain salt, you're also going to retain water. Angiotensin II, we said it reduces GFR, but it enhances reabsorption of sodium chloride and water in the proximal convoluted tubule and stimulates the release of aldosterone. Aldosterone, literally named the salt-retaining hormone, or nicknamed salt-retaining hormone. Okay. Promotes or causes retention of sodium chloride in water, reducing urine volume and raising blood pressure. Antidiuretic hormone makes us pee out less water and less salt. That therefore promotes water reabsorption. So those four are all involved with water and salt. The fifth hormone has nothing to do with water or salt. It promotes reabsorption of calcium. Who produces and releases parathyroid hormone? It's as simple as your parathyroid glands when we are hypocalcemic. Fancy word for blood calcium as well. We've talked about that before. Table 26.4 is a table from your textbook talking about those hormones, their stimulus for secretion, what they do, and their principal effects. A lot of that is straight up review from what we've learned about before, so hopefully that won't be too hard to remember. Okay. Here is a figure basically kind of walking you through what's happening in all the various spots of our nephron. Okay, I didn't talk about everything being reabsorbed and secreted, but we did talk about a good portion of those things. So you can kind of use that to help you, or not, whatever you choose. Last thing that we'll probably get to talk about today, and I say probably but we will, is storing and eliminating urine. So those other organs of the urinary system, your two ureters, your one bladder, and your one urethra. Your ureters are basically just muscular tubes draining urine that was formed by our kidneys to our bladder. Okay, they dump into the bladder actually from behind it and a little bit from below it. Okay. Together, the two ureters and the opening to the urethra form a triangular region in the bladder called the trijone. Trijone is a common site of bladder infection. Okay. Your bladder itself is basically a lot like your stomach in that it can stretch to accommodate a surge of stuff. Stomach was food and drink. Here it's just urine. Okay. We'll talk a little bit more about its role with urination. A very important layer of the bladder is the middle muscularis, called the detrusor muscle. That must contract to push urine out when we want to pee. And speaking of peeing, that involves the urethra, the tube that connects to your external urethral orifice, or your pee hole as we like to call it. Okay. What's nice is that the two sphincters that guard the urethra are a lot like the anal sphincters that guard the anus. There's an internal and there's external. Internal, just like the anal sphincter, is under involuntary control. What type of muscle do you think is making it up? I'll give you a hint. Is it skeletal or is it smooth? It's smooth because it's involuntarily controlled. So what about the external urethral sphincter, which is voluntarily controlled? It's skeletal. Same thing that we saw with the anal sphincters. Internal involuntary, smooth, external voluntary, skeletal. And at the bottom, they show you a picture. It's a sagittal section of a male and a female to demonstrate differences that exist between their urethras. This is really one of the biggest differences between men and women anatomically. Men have a much longer, about, what does it say, 5 times longer than a female urethra. And men passes through 3 kind of regions. Prostate. Prostate's something we'll talk about on Wednesday. It also passes through the urogenital diaphragm and then through the penis. It's a shared tube in men where it's only a singular tube, or a singular part of it. Not shared tube, I should just say it that way. It's kind of surprising for some people, but the vagina and the urinary system are completely independent of one another. And a lot of people don't know that. And so those are things that we'll talk a little bit more in our next lecture. Okay. Last thing that we'll talk about is the actual act of peeing, fancily known as micturition, or to micturate. Don't confuse micturition with mastication. What's mastication? Chewing. They sound similar, they're spelled fairly similar. Obviously, peeing and chewing are 2 very different things. Okay. Now, this is a reflex involving the bottom parts of your spinal cord, the sacral region, specifically the spinal micturition reflex centers of our spinal cord. And this reflex is activated when our bladder starts to fill. Remember how we talked about how the stomach fills and there are stretch-sensitive receptors in the stomach? Same thing in our spinal cord, or in our bladder. Okay. Those stretch-sensitive receptors will send signals to that sacral region of the spinal cord, okay, and then the spinal cord results with two things. Okay. Action potentials being sent to the micturition center, or parasympathetic neurons sending signals out to contract the detrusor muscle, relax the internal anal or internal urethral sphincter. Okay. At this point, a person will have the urge to urinate, but we still have, hopefully anyway, conscious control of our external urethral sphincter, which once again was skeletal muscle. Okay. For this urination to actually occur, we must send down signals from our voluntarily controlled cerebral cortex to inhibit that muscle because it's contracted. We want it to relax. It relaxes, urine can then flow out. Okay. Although emptying of the bladder is a reflex, in early childhood we learn to initiate it and stop it voluntarily. To learn control of the external urethral sphincter muscle and certain muscles of the pelvic floor, our cerebral cortex can initiate micturition or delay its occurrence for a limited period of time. So that's pretty cool. We talked earlier in the semester about several reflexes like the tapping on the patellar tendon and kicking out. That was an example of a reflex, and we mentioned this, that reflexes are involuntary and they cannot be suppressed. I lied to you. This is an example of a reflex that can be suppressed. Okay. And that's great because now we don't pee our pants. Hopefully, right? Hopefully this summer my son learns how to use this reflex or to suppress this reflex. All right, we'll call it a day here and we'll pick up with acid-base balance on Wednesday.